Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Vedika Maraj, and this lesson is brought to you by the Gauteng Department of Education in collaboration with the Sibono Discovery Center. So grade 12, today we're going to be doing the paper 1 revision program. Yes, it's come that time of the year now where you are sitting for your final examination. We hope that your exams are going well so far, and let's get right into the revision program for life sciences. Today the lesson will be presented by myself, Vedika Maraj, and as well as my colleague Agnes Moabello. Right, so grade 12s, I just wanted to say something before we go forward. Because this is just a revision lesson and we have limited time, we will not be able to go through every single topic and subtopic in detail. So I want to bring this to your attention up front so that you understand that you cannot only study what we are presenting in the revision lesson. You need to be guided by your examination guideline and make sure that you are well prepared. This is just helping you to have those little bits of information. We can give you some exam techniques and some tips and try to remind you of some important concepts. Right, so these are the weightings for the final examination for paper 1. And the lesson is going to be presented in a way where we are going to be focusing on the topics that carry more weighting, the ones that have more mark allocation, and then focusing a little bit less on the ones with less mark allocation. So the first topic that we are going to be doing is going to be responding to the environment in humans, and this is because it is 54 marks of paper 1, which is basically a third of your paper. So this is really where you should be aiming to score your marks. The second one is human reproduction, and that is 41 marks, also a large amount of the paper, basically 27%. And then the next one would be endocrine and homeostasis. Then number 4, responding to the environment. And then number 5, reproduction in vertebrates. Now, don't underestimate the mark allocation as well. Remember that these 8 marks can also be 8 easy marks, or it could also be 8 difficult marks. So we are not saying that these are the topics that you should leave out. We are just saying that focus, your most of your focus should be on the topics that carry the more weighting. So these are the resources that you have been using and that you should be using. So I want you to pay close attention. We're going to pause here for a few seconds so that you can scan the QR code. However, your teachers have also received the PowerPoint presentation of today's lesson so that you can also get the code from there. So that QR code is for the learner resources, sorry. And you will find a whole lot of information in there for you to use. Please remember, you must use your DBE study guide, your DBE textbook, past papers, your preparatory exam papers, the DBE papers over the years. All of these must be used in preparation for tomorrow's exam. You should already have started them anyway. Right, let's go right into the first topic, human response to the environment. So what does the exam guidelines say? So it talks about the brain. You need to know structure and functions of the parts of the brain. And the specific structures you need to know are cerebellum, cerebrum. Don't confuse these two as they are easily confused. They're very similar just in spelling. Cerebrum and cerebellum, medulla oblongata, hypothalamus, and corpus callosum. So you need to know structure and function of those. The spinal cord, you need to know the structure of the spinal cord and describe reflex action using a diagram. You need to know the different neurons. There are 3 different types of neurons. You need to know them. And you need to know the function of them. There are 3, the motor neuron, the sensory neuron, and the interneuron. Then you need to know the human eye, the structure and function of part of the eye, and then 2 processes, accommodation and pupillary mechanism. We'll go into them in more detail in this lesson. Then you've got the human ear. You need to know the structure and function of parts of the human ear, the process of hearing, balance, and hearing defects. Now, grade 12s, the best part, as you can see, all of these structures here are organs and different structures. The best way to study these are with diagrams. So at the back of your mind, the gap, you will find blank diagrams there that you can label and annotate so that you can have structure and function, and then it's easy document for you to study from. And reminding you that it counts 54 marks. Right, this is the brain. Okay, I'm not going to go into teaching you the different parts of the brain. However, if you get a diagram, you must be able to identify the different structures as well as provide the function. But not just that, you also need to understand the functions. You need to understand, what do we mean by the cerebrum controlling voluntary action? What do I mean by voluntary action? So they're not always going to ask you, give me the function of cerebrum. They might give you a scenario, so they can give you a specific thing. They would say that which part of the brain is going to interpret me moving my arm. Okay, I'm telling my arm that it's a voluntary action that I'm now moving my arm. Okay, and that is now interpreted by the cerebrum. So the examiner could give you a scenario. Right, so these are the parts of the brain. Please make sure you know them. Quickly take a screenshot if you don't have something like this. The three types of neurons are the motor neuron, the sensory neuron, and the interneuron. Okay, you need to know them. You need to know how to draw them. You need to know the function, structure and function. Once again, you need to know how to draw them. You need to know the direction of the impulse, very important in each one of them, the direction of the impulse. And yes, that is basically it. Okay, but the one thing that I want you to take note of is that you must only know the labels that are mentioned in your examination guideline. Okay, so that's the dendrite, the cytoplasm, the cell body with the nucleus, the myelin sheath, the axon. Okay, those are the main points that you need to know, or the main structures that you need to know. There are others in some textbooks, and those are the ones that if you write them, you're not going to get marks. Okay, so make sure you know that and you revise it. Now we move on to the reflex action and the reflex arc. Examiners like this question, okay? And it's very important that you know the difference between the reflex action and the reflex arc. So the reflex action, it's a quick automatic response to a stimulus, okay? And examples of this are the knee jerk, sneezing, and quickly removing a body part away from harm. So if you're touching a hot plate or an open flame, then your hand is quickly going to move away. That is what we call a reflex action. Now, a reflex arc is the pathway along which the impulse is transmitted. So it's the pathway, all those neurons that are forming part of the reflex that are involved in the reflex action, that is what forms the reflex arc. Okay, so like I like to ask, sometimes I like to say, okay, what is this? What is this diagram representing? This diagram over here. It's representing a reflex arc. Okay, because it's showing us the pathway along which the impulse is transmitted to bring about a response to a stimulus. Right, but then what is happening here is a reflex action. Okay, so the action that is being brought about because of this pathway is the reflex action. Now, what's the significance of a reflex action? It allows for a quick response to a stimulus without thinking to prevent damage to the body. So if you are putting your hand on a hot plate, you are not going to be thinking about it now. I'm like, oh, what's going to happen now? You're going to immediately move your hand away. And that is to prevent danger from happening, or further harm actually from happening. And please note that we always talk about an impulse, never a message. Now, grade 12s, what I need you to know. I need you to know that the pathway, you need to know this whole pathway. Okay, you need to understand that it's coming in through here, right, which is now the spinal nerve, and then we're having a branch, okay, and it's coming through the dorsal root. You need to know which neuron is this that's coming through the dorsal root. Okay, do you remember which one it is? It's the sensory neuron. Okay, so the sensory neuron is coming through the dorsal root. It's going to enter into the gray matter, and then it depends which reflex action it is, but it's going to connect with an interneuron, and then it's going to pass the impulse from the interneuron to the motor neuron, and then the motor neuron is going to enter via the ventral root, and then it's going to go to the brain for interpretation. Okay, I'm sorry, not to the brain, sorry. The motor neuron is going to go to the effector organ. Okay, so you need to know this pathway. You need to know the neurons that are involved. And you also need to be specific to what the question is asking you. Okay, let's do 1 or 2 questions and then I'll explain further what I mean. So this is a question that comes from the Free State prelim paper 1 of 2022. So whenever you get a diagram, you first need to analyze the diagram. The STEM is telling you that this is a reflex arc. Right. Now it's giving you labels here, neuron A, neuron B, neuron C. So the first thing you need to do is look at the diagram and identify the different parts. Okay. So I would identify the dorsal root, I would do the ventral root. This one, you can see the arrow is telling us that it's moving this way. And you can also see the position of the cell body, that this is a sensory neuron, right? And then the one in the middle is the interneuron. They are connecting two neurons. And then we have neuron C over here, which is the motor neuron. And we can also see because of the position of the cell body and the direction of the impulse. What is a reflex arc? Okay, so we discussed this one. It's a pathway along which nerve impulses are carried from a receptor to an effector to bring about a reflex action. Okay, I just want to stop there quickly because this is something that learners also don't understand and that they tend to forget. Is that we have a receptor, okay? The receptor is then, it's going to go from, the nerve impulse is going to go from the receptor, it's going to be carried through, right? But what does the receptor do? Do you remember? The receptor is going to convert the stimulus into a nerve impulse. The stimulus is not something that can travel along the nerve. It is the impulse that is going to travel. Okay, that's going to take the message. We don't say message, we say impulse. So we have a receptor, right, that is going to detect the stimulus. It's then going to convert the stimulus into a nerve impulse. And that nerve impulse is going to travel along the neuron and via the axon. And then it's going to eventually go to the effector where it's going to bring about a response. Okay, it's very important that we understand that, grade 12s, because that word receptor we are going to use when we are talking about the eye and the ear. And that is where the learners lose marks. 2.3.2, explain the effect on the reflex action when neuron C is damaged. Okay, so now they're saying neuron C is damaged. Now you first have to ask yourself, what is neuron C? Motor neuron, right? So if this one is damaged, what's going to happen? So do you agree with me that this sensory neuron is still going to carry out its function, right? This interneuron is still going to partly carry out its function, but it's not going to be able to give the impulse to the motor neuron, right? So that will mean that the person would be able to feel the sensation because the sensory neuron is still doing its job. However, they will be unable to react to the stimulus because now the motor neuron cannot take the information to the effector organ to bring about the response. Name the disorder that is the result in the breakdown of the myelin sheaths of neurons. Yes, this is a disorder that you need to know. There are two disorders that you need to know for the nervous system. It's Alzheimer's and it is multiple sclerosis. So the one that degenerates the myelin sheath is the multiple sclerosis. Okay, you also need to know the symptoms of these two. You need to know what they are. You need to know what causes it. You need to know the symptoms of it. Okay. And they can ask you that question in different ways. They can give you a case study. They can give it to you in this way. It can be in a multiple choice. It can be in a biological terms. Okay, but it can be anywhere in your paper. So this one was last year's question paper. It was a diagram representing the reflex arc for the knee-jerk reaction containing only one synapse. The arrows indicate the transmission of nerve impulses. Describe the pathway of the impulse in this reflex arc to bring about the knee-jerk reaction. So as you can see over here, this one is telling us, it's giving us some information. It's giving us the patella tendon, okay, which is now found in the knee. And then it's giving us a synapse that happens here. And then it's giving us this neuron that is going to the quadriceps muscle, okay, which is the skeletal muscle, right? So you can see here that this is still a sensory neuron. How do I know it's a sensory neuron, grade 12s? Because I'm looking at the direction that it's showing us and I'm looking at the position of the cell body. Okay? But now this synapse, they told us here, only one synapse occurred. So it's very important. I know that in class they've taught you it goes from the sensory neuron to the interneuron to the motor neuron. However, not all reflex actions follow this pathway. In this particular one, there's no interneuron. So there's no connection between, there's no interneuron. So the connection is purely between the sensory neuron and the motor neuron. So in your answer, you need to talk about all of these points and not talk about an interneuron because there is no interneuron here. So you say the impulse is transmitted from the receptors in the patellar tendon through the sensory neuron and the synapse to the motor neuron and then to the quadriceps muscle. Simple. They just want you to describe the pathway of the impulse. Are you all fine with that? Okay. Now, hopefully you have realized, oh my word, Verika's already gone on to the eye, and what happened to the sympathetic nervous system and the parasympathetic nervous system, autonomic nervous system, and I didn't talk about central nervous system. Please take note that you still need to study those things. Like I said, we don't have a lot of time to go through everything, so we are only focusing on what we can. So the structure of the eye, these are 2 slides. Please make sure you know the structure of the eye and you know the functions. Examiners love to ask you to compare the different functions or compare the structural suitability or compare the structures. Okay, so they like to ask you if there's something similar. For example, the sclera is made up of a tough white inelastic layer, whereas the lens is made up of elastic, transparent, biconcave structure. So they like to take things that are a little bit similar but different, and they like to ask you to compare it. So just make sure you know your stuff. Okay, make sure you know your structures, make sure you know the function. Both of these diagrams are excellent for you to use when you are studying. Once again, remember that you can also use Mind the Gap, where they have in 1 pages. We've got so many resources in that link that you can use to study. Now we move on to the pupillary reflex, okay, the pupillary mechanism. Another question that examiners love. And I want to just pause a little bit here so you can see what's happening. Do you see how the pupil is dilating and constricting there? Okay, so it's pretty cool. And now you are going to have to describe in the exam, maybe you're going to have to describe in the exam how this is actually happening. Okay, so the pupillary mechanism is in a response to the amount of light. Okay, so firstly, we need to understand, I need to remind you that the iris is made up of 2 muscles. It's made up of circular muscles and radial muscles. The circular ones are the ones that are circling the pupil, and the radial muscles are the ones that branch out, okay, or that radiate out. So when there is bright light, we need light to see, okay, but too much light can damage the retina. So when we have bright light, we want to want our pupil to constrict so that we can restrict the amount of light that is entering the eye. So in bright light, the circular muscles of the iris are going to contract, the radial muscles are going to relax, and the pupil is going to constrict. And the amount of light entering the eye is reduced, so less light enters the eye. Okay, now there's a few points that I want you to remember here before I go tell you about the tips. When you are talking about the circular muscles, you need to mention that they are in the iris, that they're the circular muscles of the iris, because there are other circular muscles in the body, right? So you need to say it's the circular muscles of the iris. Then you can see here in this little description, we have bolded for you certain words that can help you remember. Okay, and these words are bolded and they are put into this little thing over here that you can use, CCRRPCLL. So this stands for circular muscles contract, radial muscles relax, pupil constricts, less light enters the eye. Perfect, right? Now, another way to help you remember this is that you can learn bright light, and then if you are asked dim light, you should know that dim light is the opposite of what happens in bright light. So let's have a look at the dim light one. In dim light, the circular muscles of the iris relax. I'm also highlighting iris there to remind you. The radial muscles contract, the pupil dilates, and the amount of light entering the eye is reduced, or more light enters the eye. Let's once again look at our little thing over here. We have the circular muscles relax, the radial muscles contract, the pupil dilates, and more light enters the eye. Okay, so now you've got quite a few tips to remember this. But grade 12s, let me warn you about something. They're not always going to give you they're not always going to ask you describe the pupillary reflex in dim light or in bright light. They could give you a scenario. They can tell you that somebody is playing outside, you're playing outside with the ball, and it's a very bright day, the light it's bright, and then you're coming indoors where the light is dimmer. You need to realize that the light is now dim inside, and that you need to talk about dim light. Okay, now moving quickly into the accommodation. Accommodation is the focusing of light on the retina. So accommodation is different from pupillary mechanism. Pupillary mechanism, the stimulus is light. It's controlling the amount of light that enters the eye. Whereas in accommodation, it is still light, yes, but the function is that it's helping you focus on whether an object is far or near. Okay? Mammals focus by changing the shape of the lens. So the lens is going to change in shape. The lens is less convex for distant objects and it's more convex for near objects. What do we mean by convex? Okay, so the lens is actually biconvex. Bi meaning two. Convex, okay, is the shape. So this shape over here, if you look at this angle, this part over here, this is convex. So if it's biconvex, there's two of these that are together. Okay, so when it becomes less convex, that means it's going to become thinner, you could say, or flatter, but we don't like those words. Okay, you must say less convex. So you can becomes less convex or it becomes more convex. And you see more convex and less convex, and it's normally convex. Okay, so I like to use that example with the hands to help me to understand, but also look at this one. Look how cool it's going. See, it's convex, more convex, less convex, right? Now we come to the words, right? Now, also diagrams. I love diagrams for this because it can really help you to understand. So let's go through it quickly. When the object is more than 6 meters away, what's going to happen? So more than 6 meters away means that it's far vision. The ciliary muscle is going to relax. When the ciliary muscles relax, the sclera is pulled backward and the suspensory ligaments are taut. Okay, then the tension on the lens increases. The lens becomes less convex because now it's taut, right? So when it's taut, it's going to be pulling the lens and the lens is going to become less convex. The refractive power of the lens decreases and a clear image is focused on the retina. Okay, so far vision, less convex. Far vision, less convex. If it is less convex, it means that the suspensory ligaments were taut. If they do not contract and they do not relax because ligaments are not muscles. Okay? So you must use the correct words. They become taut or they slacken. When they become taut, they are pulling. So then the lens is going to become less convex. Now, when the object is less than 6 meters away for near vision, that means it is near. So now we've got more convex. Near vision, more convex, right? And it is the opposite of far vision. So when you are studying this grade 12, there are different ways to help you remember it. Me, I'm a picture person. So for me, the way that I'm reiterating this with the diagram and with actions, that's how I study. But we are all different. If you are having a problem to study and you know that the life sciences is a lot to study, learn far vision or near vision. And then you know that if you get asked the other one, then it is the opposite of the one that you know. Okay, so let's quickly go through that one. The ciliary muscles contract, the sclera is pulled forward, the suspensory ligaments slacken, the tension on the lens decreases, the lens becomes more convex, and the refractive power on the lens increases, and a clear image is formed on the retina. We have seen questions where they've asked you to draw this, so make sure that you understand what is happening. Okay, this is just a diagram a table that gives you the difference between accommodation and pupillary mechanism, the different things that happen, the different parts that are involved. Then we have a quick question. Here we're going to identify the parts. We have A, yes, that is the blind spot. We have C, the cornea, and we have D, it's the sclera. The sclera, the outermost layer over there. Describe how the muscles in the iris enable a person to see in dim light. So this was a straightforward one, right? They literally said, how are the muscles in the iris enabling the person to see dim light? So now you mean dim light, it means that the pupil needs to dilate. Okay, so the radial muscles contract, the circular muscles relax, the pupil widens or dilates, and more light enters the eye. So in this instance, they didn't talk about iris because iris was in the question, but you must always train yourself that when you are answering questions on pupillary mechanism, always mention the iris. Name the process that occurs in the eye when a person focuses on objects at different distances. Okay, that is called accommodation. So when we're talking about different distances, it's accommodation. When we are talking about the amount of light, it is pupillary mechanism. Explain how the shape of part B enables a person to read a book. So now we go to part B. What is this? It is the lens. Okay, if you are reading a book, it is near vision. Okay, so what did I say about near vision? Near vision, more convex. Near vision, more convex. Suspensory ligaments, slacken. If the suspensory ligaments are slackened, that means the ciliary muscles are going to contract. Contract, slacken, more convex. Okay? So let's look if we are correct. It's more convex, and they ask what's happening to the shape. So the direct answer is that it is more convex. You need to explain. That's the reason why you need to say light rays are refracted more to focus on the retina. Okay. Because they want to know how the shape of B enables a person to read a book. So you have to say, what is the shape? It's convex. You need to explain how. And it's because we have more diffraction taking place because of that more convex structure. Okay. Refraction means the bending of light. Okay, now we move on to the ear. Now the ear, same story, structure and function. You need to know the structure and the function of all the parts of the ear. And there's a lot of words there, grade 12s. I know there's a lot of confusing words there. Diagrams, once again, blank diagrams, mind the gap, 1 pages. Make sure that you have them. Then the path of sound. This is also one that the examiners like to ask. And if they ask you to describe the pathway of sound, that's really nice, 7 marks, okay? But learners lose marks because of 2 reasons. Number 1, they don't always use the correct terminology. Number 2, they are not using the correct stimuli. Okay, and then the impulse. So we're going to go through it. What you must remember is that we have pinna. Okay, we have the outer ear, middle ear, and the inner ear, right? Then we've got the pinna. What is picked up by the pinna? It's a sound wave. That sound wave is a sound wave until it reaches the tympanic membrane. When it reaches the tympanic membrane, it becomes a vibration. It hits the tympanic membrane, the tympanic membrane vibrates. It becomes a vibration. That stimulus is now a vibration. That vibration is going to cause the ossicles to vibrate. Okay, so now this is called a vibration all the way as we go through the oval window. Once we leave the oval window and we enter the cochlea, we are now entering a space where there is fluid. So now we don't have a vibration. Do we say that fluid vibrates? No, it is now pressure. Okay, so now we've got pressure waves. And then we reach the organ of Corti, which is a receptor. Very important. In the eye, the receptor is the what? In the eye, the receptors are the photoreceptors. We didn't mention that just now. They are the photoreceptors, which are the rods and the cones. Here, the receptor is the organ of Corti. Okay, learners forget that. And they love to ask that question. What is the receptor in the ear? And then it's the organ of Corti. Okay, that receptor is going to convert the stimulus. These pressure waves are now going to be converted into a nerve impulse. And this nerve impulse is going to be taken by the auditory nerve. To where? Because we want to sensations. Where are the senses interpreted? The cerebrum. So you need to know that it goes to the cerebrum. That is the pathway of sound. Structure. Know the structure, know what the stimulus is, know what the receptor is, know what the receptor does, know which part of the brain it interprets sound. There are 2 ways the inner ear regulates balance. Okay, now this question, they also love to ask it, usually 5 marks. Learners lose marks, and I don't understand why. Okay, let's quickly look at it. So, there are also receptors here. But now, the only difference here is that there are 2 different receptors. And the receptors are both responsible for balance, but the stimulus is a little bit different. So when the crista is stimulated, it's because we have a change in the direction and the speed. So you are running. Okay, you are running and then you turn the corner or you turn around. That is changing in the direction and speed. Okay, and that receptor is going to be the crista. And the crista is found in the semicircular canals by the ampulla. But when there's a change in the position of the head, so now I'm like this, and then I'm bending my head forward, or I'm moving my head that way, that is the change in the position of the head. And that receptor is picked up, that receptor that is involved there is the macula. And that you find in the sacculus and the utriculus. Okay, so there's a little thingy over here to help you remember. Okay, but I remember it. Everybody will have a different way. Okay, but as long as you understand which receptor, because when the examiner is going to ask you, if they just ask you how is balance maintained, okay, then you're going to talk about the crista and the macula. You're going to say all of this. You're going to say when there's a change in direction and speed, the crista is going to be stimulated. When there's a change in the position of the head, the macula is going to be stimulated, you see. But the problem comes in when the examiner gives you a specific situation. They tell you somebody's running a race or they're doing something, and then you're going to need to ask yourself, is this direction and speed, or is this position of the head? And that's where it's going to make a difference on which receptor you mention. Okay, but the good news is that this part is the same. Whether you're talking about the macula or the crista, this part is the same. Both receptors convert the stimulus into an impulse and send it via the auditory nerve to the cerebellum. That's very important. It is the cerebellum. The cerebellum is the one that is going to maintain balance, which sends an impulse to the skeletal muscles to maintain balance. Okay. So like I said, this is the part that may differ depending on the scenario they give you. If they don't give you a scenario and it's a straightforward question, you mention both, but this part will remain the same. Okay, and quickly describe the role of the ear in maintaining balance. We just spoke about this. Like I told you, you mentioned both. Crista are stimulated by the change in speed or direction of movement of the head, and the macula is stimulated by the change in the position of the head to generate an impulse, which is transmitted by the auditory nerve to the cerebellum for interpretation. Right? Nice 5 marks. That is the end of the nervous system. That was a mouthful. Okay, but very important section. 54 marks. Grade 12, make sure you know how to answer questions on the nervous system. I just want to also mention something. All of these topics that we are going to discuss with you, we're not going to do a lot of activities, but you must remember that any topic in life sciences, they can ask you anything. They can give you a diagram, they can give you an investigation, they can give you a case study. Okay, it's not necessarily that you're only going to see the picture of the eye to get a question on the eye, or the nervous system is not only subjected to drawings that they're going to give you. They can ask you a topic in any way. Okay, so the best way to do this is just know your work and keep your wits about you, read the questions carefully so that you can answer them to the best of your ability. And do a lot of past paper questions. That is going to help you to expose yourself to the different types. Human reproduction. Okay, also quite a few marks, 41 marks. Okay, it's still a lot. So this is showing you all the things you need to know. Like I said, we're not going to go through all of them today, but this is what you need to know. Please make sure take a photo quickly. I'm going to pause for a couple of seconds that you can take a photo, and then you need to know female reproductive system, male reproductive system, puberty, menstrual cycle, hormonal control of the menstrual cycle, development of the fetus. Okay, make sure you know everything about this topic. They love this topic. Right, male reproductive system. You need to know how to label a diagram of the front view and the side view. Okay, because you never know which one they're going to ask you. And as you can see, they kind of look different from the side and the front. Okay, so you need to know the different parts. That's why I showed you this diagram. Okay, make sure you know them. Make sure you know the functions of the different parts. Make sure you also know, in life sciences, they don't generally ask you, give us the function of the vas deferens. They will tell you, if the vas deferens is damaged, how does it affect fertility? That's how they're going to ask you questions. So then you must ask yourself, okay, what is the function of the vas deferens? The function is to transport the sperm cells. So if it is damaged, it cannot do that. And if the sperm cells cannot be transported, they cannot be released from the body and fertilization cannot take place, which can lead to infertility. Okay, so in life sciences, they don't always ask you the function straight out, but you need to know the function in order to apply yourself to the question that they're going to give you. Then we have the female reproductive system. Also, you need to know the front view and you need to know the side view. The front view is like a little lampshade, and then the side view, okay, it's looking similar to the male over there. You just need to know the different parts and you need to know the functions once again. Then we move on to gametogenesis. Gametogenesis is the formation of gametes by meiosis. In females, this is called oogenesis, and the gamete in females is the ovum. And in spermatogenesis, that is occurs in males, and the gamete we are talking about here are the sperm cells, okay? And they are both haploid. Now, grade 12s, I want you to know the examination guideline description of this by heart. Okay, it was asked, I think it was in the prelim paper where one of these questions was asked, and the marking guideline was straight from the examination guideline, and learners got it incorrect. Okay, don't underestimate how much information is in your exam guideline. So this is what you need to know. You need to know the description of oogenesis and spermatogenesis. You need to know both of them, and you get it from your exam guideline. Once again, take a picture quickly if it's going to be an easier form of reference for you to learn from for tomorrow. Right? Gametogenesis, oogenesis, and spermatogenesis. You need to know them. Okay? And also understand them. Understand what is happening here. Okay. I think I must just mention that quickly. The main things we must understand here is that we need to know the hormone that is involved here. It's FSH here. It's testosterone here. We need to know that meiosis is going to be occurring. Very important. Okay, that meiosis is occurring here, because learners get confused. And actually with oogenesis, we are first undergoing mitosis, because we have these diploid cells right in the ovary, and they are going to undergo mitosis to have numerous follicles. Okay, then that's what happens before puberty. And then on the onset of puberty, because of FSH, that is when one cell is going to enlarge and undergo meiosis. Okay, so it's also important for us to have the understanding about this, because might be required to answer questions on that. So here we have a scientific investigation. Like I said, they can ask it to you on anything. Okay, so let's just recap. What do we always say about scientific investigations? You need to read through all the information very carefully, and then you need to think about the following things. Okay, you need to think about the aim. What is the aim? From the aim, you're going to get your two variables. You're going to get your independent variable, and you're going to get your dependent. Okay? You're going to get your independent and your dependent variables. Then you are also going to ask yourself, are there any constant or fixed variables? Then you're going to ask yourself, how did they ensure reliability? How did they ensure validity? Is there maybe a conclusion? So while you are reading this, your mind must already be starting to think about these things. Okay, so how do you identify? Okay, first let's read it. Then we're going to identify these things. Scientists conducted an investigation to determine the relationship between age and fertility in men. Now I'm seeing here to determine, so that means that this is my aim. This investigation was done from 1999 to 2017. Wow. Is that not a long time? How many years is that? That's like 18 years. Okay? So when you see values in a scientific investigation, usually it's going to be giving you some information that's related to reliability and validity. They used 1,294 healthy men from the age group 16 to 65. Men with occupations working in environments involving exposure to excessive heat were excluded during the selection of participants. Okay, so they don't want people that were exposed to excessive heat. What are they doing again? They are checking a male fertility. Good. The procedure was as follows. Semen was collected after 5 days of no sexual activities. A specialized microscope was used to determine the sperm count, which is the number of normal sperm per milliliter of semen, and progressive motility, which is the ability of sperm to swim effectively in a straight line. An electron microscope was used to determine sperm necrosis, which is immature dead or dead sperm per fresh semen sample. Okay. So let's check. What are the questions? Identify in this investigation the independent and dependent variable. Now let's quickly go back to the aim. So they're telling us determine the relationship between the age and the fertility in men. Okay, so which one is the independent and which one is the dependent? Right, the independent is the age. Yes, it is the age because the age is a given. The scientist is the one who decided what age group they're going to use. Okay, so the age is the independent variable, and the dependent variable is the fertility because the fertility is dependent on the age of the men. Okay, state 3 ways in which the dependent variable was determined. So now they're saying, yes, you know the dependent variable is fertility. How do they determine the fertility? And it's got to do with the semen, okay? It's got to do with the sperm count. They determined the sperm count, the progressive motility, and the sperm necrosis. Okay, they took into account 3 things. The sperm count, and they explained it to you. You can even put the explanation there if you wanted to. But if you just said, because they looked at the sperm count, the progressive motility, so that's looking at the speed of how fast they swim, or they move, and then sperm necrosis, which is what they say is the immature dead sperm. Okay, so that's how they determined infertility. It's not only determined on one thing, it was determined on these 3 things. Now, grade 12, where do you get that information? Yeah, they told us the procedure is as follows. Okay, so a lot of the information they're going to give you for your scientific investigations are going to be in the information given to you. Give 2 reasons why the investigation can be considered reliable. So remember I said you look for the parts where there's numbers. Okay. So you need to ask yourself, what makes the investigation reliable? It's having a large sample size, okay? And it is repeating the investigation or doing the investigation over long periods of time. So you cannot ask the question. They want to know in this investigation, why can it be considered reliable? If it is asked to us in the past tense, that means it's something that they told you happened. So you need to go back to the paragraph. Okay. And in the paragraph, they told us that it was done from 1999 to 2017, which we said was 18 years. That's a very long time to conduct an experiment. And then it was also a large sample size was used, which is 1,294 men. So you're not going to say the generic answer. You're not going to tell me a large sample size was used. They did it over a long period of time. You are going to give me specifically what was given to you in the information. Explain why men with occupations involving exposure to excessive heat were excluded from the investigation. Beautiful question. Why did they give us that piece of information? Why is it important? We're looking at male infertility. And what does excessive heat exposure, what does it do to sperm? Do you remember? Right? So high temperatures can affect fertility. Okay, remember that when we are giving answers in life sciences, remember that when it's an explained question, you don't always have to give the explained question step by step the way it gives you in marking guidelines. Okay, you need to understand what is happening. So what does excessive heat do? High temperatures can affect fertility. So therefore, they did not use these people because that age can be the only independent variable. Because they want, age can also affect, age is affecting, they want to figure out with how age is affecting fertility. But if they're going to include another variable of exposure to excessive heat, that can also affect fertility. So they want to exclude that so that they can see that only age is the one that is going to be the variable that is going to affect the result. Okay, therefore decreasing the validity. So if they are going to have people that you're going to add that variable of excessive heat, then we are going to be decreasing the validity. Why? We are going to be making it now less fair, because now we don't know, did the infertility happen because of the age, or did it happen because of the exposure to the heat? So it's a tricky way of asking us to talk about control, né? The control of the experiment, or actually validity. A tricky way to be asking us about validity and fairness. Right, now we move on to my favorite, the menstrual cycle. Okay, now when it comes to the menstrual cycle, grade 12, the very important thing about the menstrual cycle is that you need to know the hormones. You need to know the glands, you need to know the hormone that the gland secretes, and you need to know the function of the hormone. Okay, this table is very, very NB. And FOLP is helping you to remember that it's FSH, estrogen, LH, progesterone. And they are going in the menstrual cycle from day 0 to day 28 in that order. We first have FSH secreted, then we have estrogen, then we have LH, then we have progesterone. You need to know which one secretes what. The pituitary gland secretes FSH and LH. The ovary secretes estrogen and progesterone. Which parts of the ovary am I talking about? Very important. Estrogen is secreted by the developing follicle and the Graafian follicle. The bigger the follicle, the more estrogen it's going to secrete. Then the progesterone is secreted from the corpus luteum. The corpus luteum can only be formed once ovulation has occurred. Ovulation can only occur when the Graafian follicle is mature. And then because of that LH, the Graafian follicle is going to rupture and it's going to release the ovum. That empty Graafian follicle is then going to be converted into a corpus luteum. Okay, only then is progesterone going to be secreted. And once gestation occurs, the placenta is then going to secrete the progesterone, and that is going to be maintaining the thickness of the endometrium during pregnancy. Okay, so you need to know these functions. One thing that I want to, let's quickly go through it because it's very important. The pituitary gland, we said it secretes FSH. What is the function? It stimulates the development or the growth of the follicle. Okay. LH stimulates ovulation and it stimulates the conversion of the empty Graafian follicle into a corpus luteum. Estrogen, the thickening of the endometrium. It's building that lining, the endometrium. We don't say wall. We don't say endometrium wall. We don't say uterine wall. We say endometrium. Then the progesterone maintains the thickness of the endometrium. It maintains the thickness. This one is building it up. This one is maintaining it. Okay, and then we don't say maintain pregnancy here. That is not the primary function of progesterone. If they're giving you a question and they're telling you in the question, they're giving you some indication that pregnancy has occurred, then we can say that it's maintaining the thickness of the endometrium for pregnancy, okay, or during pregnancy. Then we have the last one, and then we spoke about that. It maintains, then we can say it's maintaining the pregnancy by maintaining the thickness of the endometrium. Right, then we go on to the menstrual cycle. Please make sure you know how these are all interacting with each other. You need to know the hormones and you need to know how they're interacting with each other. So this is the ovarian cycle. What's happening in the ovary? Okay, so you need to know what's happening in the ovary. You need to know how the Graafian follicle is developing, what causes ovulation to occur, which day does it occur on, ovulation on day 14, and how we are going to progress into the corpus luteum. Okay, and then you also need to know this one, which is the uterine cycle. Okay, you need to know the uterine cycle, and that is how the endometrium is shedding during menstruation. That's this part over here, from day 1 to day 7, and then how it's going to start building up over here, and then how it's going to be maintained. Okay, later on, and then if fertilization does not occur, then it's going to start decreasing again. Now, let's quickly talk about the hormonal interaction. The first thing that's going to happen is going to be FSH is going to come in here. Okay, from the pituitary gland. It's going to travel via the bloodstream. It's going to enter the ovary, through the blood vessels in the ovary, and then it's going to cause the follicles to develop. And as the follicles are developing, that's the reason why we have an increase here of FSH. It comes from the pituitary gland. So when it's going to do that, these follicles are going to secrete estrogen. That's why estrogen, which is the red one, is going to start increasing. Can you see that? Estrogen is increasing, it's increasing because FSH is increasing, and as the follicle gets bigger, it's going to secrete more estrogen. The bigger the follicle, the more estrogen is going to be secreted. And when it does that, we're going to have the building of this endometrium. Okay? The endometrium is going to become thick. Then, when estrogen is going to surge, when we have the surge of estrogen, that is going to stimulate LH to surge. And when that LH peaks, that is when ovulation is going to occur. The Graafian follicle is going to rupture. It's going to release the ovum, and the ovum is now sitting in the fallopian tube, right? Waiting to be fertilized. But life is carrying on inside here, right? We need to know what's going on here now. But remember that the ovum is now in the fallopian tube. Okay, so now day 14, that has happened. Now the LH is going to decrease a bit. The estrogen is going to decrease a bit because now this Graafian follicle is empty. Okay, then LH is now still going to be working to convert that empty Graafian follicle into a corpus luteum. This is the corpus luteum over here. And that corpus luteum is going to secrete the progesterone. So can you see the progesterone is now increasing, and that causes the endometrium to become thicker, to maintain the thickness and become more thicker, more glandular, more vascular. Okay. Now, remember I told you the ovum was there in the fallopian tube. Now, if that ovum does not get fertilized, okay, if it does not get fertilized, then that means we're not going to need this endometrium anymore, because implantation is not going to occur because fertilization did not occur. That is when the corpus luteum is now going to start shrinking. It's going to start degenerating. We talk about degenerating. We talk about disintegrating. We talk about shrinking. We don't talk about dying. Okay, the corpus luteum does not die, we say that it degenerates or disintegrates. And when it is doing that, it's going to become smaller. Now, just how I told you, as the Graafian follicle develops and becomes bigger, it secretes more estrogen. So as the corpus luteum is going to become smaller and degenerate, it's going to secrete what? Less progesterone. So when it secretes less progesterone, there's nothing to maintain the endometrium. And that is why menstruation occurs and the endometrium is shed. Now, let's back it up. Go back to day 14. Once the ovum has been released, now it's sitting in the fallopian tube. Now it gets fertilized. Okay, now it gets fertilized. Now what's going to happen? Implantation has to occur, right? Which means that we need that endometrium, which means that we need progesterone, which means the corpus luteum cannot degenerate. It has to stay intact so that it can carry on secreting lots of progesterone to maintain the endometrium lining. And that's when this graph will look a bit different. The progesterone will still be high and the endometrium will also still be thick if pregnancy occurs. Okay, this is another way to show you, some learners like flow diagrams. This flow diagram is also explained to you what is happening in the menstrual cycle. You can also quickly take a photo of it. It is also in the DBE study guide. Right, we're going to quickly do a question about Sheehan syndrome. Sheehan syndrome is a condition that results in females having very low levels of luteinizing hormone. The graph below shows the hormone levels of two different women during a 28-day menstrual cycle. Woman A has normal luteinizing hormone levels, while woman B suffers from Sheehan syndrome. Okay, so here they're showing us the hormone levels. They're showing us LH. Woman A is normal. Okay, let's just remind ourselves. You should be doing this as well, grade 12, when you get things. B is Sheehan's. So this one has less LH. Right? Yes. So LH, we have this dotted line over here is you can see over here around day 14 we have a surge. Okay, so normal. But here we have in woman B, can you see that the LH levels now are remaining constantly low? Then we have another woman there. This is woman X. Hormone X, sorry. This is hormone X in woman A. So here we still have woman A. Remember, the woman has more than one hormone. So LH is low, but now hormone X, what is happening? As you can see, to me, it is following a normal curve, okay? Because this one is probably what? Progesterone. Why am I saying it's progesterone? Because it is increasing after day 14. And as you can see over here, well, this one is showing us it's like sort of being maintained, right? The level. Okay, so let's look at what questions they ask us. State two functions of LH during the menstrual cycle. Yeah, now you know this because we just did it. It stimulates ovulation and stimulates the formation of the corpus luteum. Besides LH, name one other hormone that is secreted by the pituitary gland during the menstrual cycle. So we spoke about LH, the other one is FSH. Give the name of hormone X. So we spoke about that. It is progesterone. Okay? And why? Because, like I said, it increased after day 14, right? Because it's from the corpus luteum. Use the information in the graph to explain how level of hormone X will be different in woman B. Okay, so they're saying in woman A, that's what's happening to it, right? But now explain how it would be in woman B. So now you ask yourself, what is wrong with woman B? Woman B has very low LH. And what is the function of LH? The function of LH is to stimulate ovulation. Okay, it causes ovulation. So if the LH does not peak, ovulation cannot occur. Okay, and so that is the reason why. And if ovulation cannot occur, the corpus luteum will not be formed. And if the corpus luteum will not be formed, then the progesterone will not be high. So in this person, the progesterone will be low. So do you see how we get to the answer? You ask yourself first, what is the problem with woman B? Okay, it's a low LH. What does that cause? Okay, what does that cause? And that's how you eventually get to your answer. And then you write it down exactly how you're saying it. The progesterone levels are low. Why? Because the LH levels are low. Ovulation cannot take place and no corpus luteum will develop. You see, simple. You can even start here. You can even start at this point and end with this point. You're still going to get your marks. Okay. What evidence in the graph suggests that woman A is pregnant? Now, this is woman A. Now, can you see what's happening to the progesterone? What did I tell you earlier about the progesterone? If fertilization did not occur, then around day 21, the progesterone levels are going to decrease. Okay? So that menstruation can happen on day 28. But what's happening here now? From day 21 all the way through to day 28, and probably beyond, we have progesterone levels remaining high. Therefore, the woman is probably pregnant. And all you need to say is that progesterone levels remained high. Okay, lovely menstrual cycle question. Like I told you, they can ask you anything. They haven't asked the menstrual cycle in the end of the year for a while. But now, if they do ask it tomorrow, you'll be prepared. Okay, development of the zygote, implantation and gestation. The things important, this is a quick part before I hand over to my colleague. Very important, fertilization. Remember we spoke about that ovum that's in the fallopian tube. Okay, now when we get to gestation, grade 12, don't forget where that ovum came from. Remember, we still have a menstrual cycle that's happening. The menstrual cycle's function is to prepare the body for possible pregnancy. Every month it's possible for a female to fall pregnant in the puberty, once they've reached puberty. Okay, so what is fertilization? It's the fusion of the haploid sperm nucleus and the haploid ovum nucleus to form a diploid zygote. We've got a haploid, which is half the number of chromosomes. We have another haploid, half the number of chromosomes. They're going to fuse to form diploid, which is the full set of chromosomes, and then that is a zygote. Sorry, we just have a little technical glitch. Can we just pause for 30 seconds, please?
I want you to ask yourself these 3 questions. Number 1, what do I want? Do I want to become an A student or do I want to become an average student? Number 2, I want you to ask yourself, how do you plan on getting there? Are you going to put more extra hours or are you going to just sleep? Number 3, who will be with you in this journey? because it is important that we associate ourselves with students who have the same vision as us, who want to become a student, who want to put in the extra work, who want to become better at what they do. Unsuccessful people do not plan, they just let life happen to them. But successful people,
Welcome back. Sorry about that technical glitch. Let's move right on with it. So that is fertilization. Now we've got the zygote. Now, what's going to happen to this zygote? It's going to develop, because that zygote needs to develop now into an embryo and develop into a fetus and then eventually into a baby. Okay, so how is this going to happen? So remember we spoke about the egg, okay, the ovum, sorry. We have the ovum over here. Then we've had fertilization that occurs. I explained to you. You also need to know the process of fertilization, about how the sperm makes contact with the ovum, etc. We don't have time for that now, though, to go through it. And then we have the development now. So once it becomes a zygote, okay, once it becomes a zygote, so we have a zygote, then this holds everything that's going to occur here is going to be growth and development. And this is all happening because of mitosis. So then it's going to undergo mitosis, the zygote, to form a 4-cell structure, an 8-cell structure, then a morula. A morula is a ball of cells. And a blastocyst is a hollow ball of cells. So this is going to develop via mitosis, not meiosis, mitosis. And then it's going to go down into the uterus and it's going to implant. We have implantation that is going to occur. It's going to implant into the endometrium. Okay. And once it does that, grade 12s, it's going to form different layers. Okay, and the outermost layer that the structure is going to form is called a chorion. And that chorion has chorionic villi. And those chorionic villi are going to embed into the endometrium. Okay, and it's going to embed into the endometrium, which is the maternal tissue. Okay, and then so we have the outer layer, which is the chorion, and that chorionic forms chorionic villi. That chorionic villi plus the endometrium is going to make up the placenta. That's what this diagram is showing us. Okay, and then it's also showing us that we have an inner layer, which is inner membrane, which is called an amnion. We have an amnion sac, which contains amniotic fluid. Okay, and then we also have an umbilical cord, which is going to extend from the fetus to the placenta. You need to know what the function of the placenta. You need to know all the functions of the placenta. You need to know how the fetus gets nutrients. You need to know how the waste products are excreted from the fetus. You need to know all of that. This gives us a little summary of all the different structures. You need to know this diagram as part of the exam guideline. You need to know the development of the fetus in the uterus. So you need to know all these structures. You need to know the functions. Please make sure you know it. You need to know that the umbilical cord have 2 arteries and a vein. You need to know the functions of them and all of these that are summarized for you over here. Okay, so quickly recap, grade 12. I know we did a lot there. Please, just to remind you that we do not have a lot of time to do everything, but please make sure that you go into everything properly. And now I'm going to hand over to my colleague Agnes, who's going to take through to the last 3 topics. That's all from me. Thank you.
Good afternoon, grade 12 learners, and welcome back to our last session. Right, I'm going to take you through endocrine system and homeostasis. And my name is Agnes Mwabelo. Right? So with the endocrine system, girls and boys, you need to know the functions of the endocrine glands. You need to know the location where they are situated, and you also need to know their secretions, what they're secreting. Also, we'll be looking at homeostasis, which is most problematic when we are asked questions as grade 12 learners. So with homeostasis, remember with all the definitions, also you need to look back at your exam guideline. So homeostasis is a process whereby it maintains the constant internal environment despite there are changes externally or internally. So endocrine system carries 34 marks of our paper 1. Yes, girls and boys, we are now counting hours before we can write our paper 1. And we are thinking of you. Have that courage. Everything that you have studied, you're going to make it. I am certain of that, right? Now, for negative back mechanism to happen, remember we say it leads to what? To homeostasis. And remember with homeostasis is what? Is the process of maintaining what? The constant internal environment, despite or regardless of the changes that are taking place externally or internally, right? Now, there are 3 main components for negative feedback mechanism to take place. So we're talking about the receptors, we're talking about the control center, we're talking about the effector. Remember, because we are talking about the endocrine glands, they are secreting hormones, by the way. Right? So with the receptors, what are the receptors? Now, receptors, we know they are detectors. What are they detecting? We're saying they are detecting the imbalance of the hormones, right? So if the hormones are not balanced, we are going to talk about the receptors that are going to detect that. Now, with the control center, it controls the balancing of the hormones. Remember, receptors, they detect the imbalance, and then the control center controls the balancing of the hormones. Come to the effectors. Effectors, they respond to the imbalancing of the hormones, right? Then, coming to the endocrine glands, remember you need to know or remind yourself. Remember, with this session, we are just recapping on what we have studied throughout the year. So with the endocrine glands, girls and boys, you must know the location. Where do we find them? You must know their secretions. Now, according to exam guideline, also, you must know the definition. Definition of what? Of hormones. So hormones are what? Chemical messengers. And remember, we've got types of endocrine glands there. You know their names, you know what they secrete. Where they are secreted, they are not going to function there. Hence, we are saying hormones are chemical messengers that are secreted directly into the blood. Because we're talking about the endocrine, don't forget again, endo means inside. So the endocrine glands secrete their secretions. Remember, they also are ductless. They do not have ducts. So they secrete the hormones directly into the bloodstream. And I've mentioned that they are secreted elsewhere, but they are going to be functioning, or they are going to be targeting specific organs. So hormones are secreted directly into the blood, but where to? To the target organs. Okay? Now, what are the factors that must be kept constant? Remember, we're talking about homeostasis in plants, in the blood, and also in our body cells. So there are factors that must be kept constant all the time. So remember, we are talking about secretion of hormones. So the body cells do not need too much of secretions of hormones or less. Hence, we're talking about the maintaining of constant levels internally. So right? So what are those factors that must be kept constant in the body cells? We talk about the thyroxine level should be kept constant all the time. We talk about the glucose, it must be constant all the time. The salt, we're talking about the water, we don't need more or less. Everything must be constant or must be maintained in that way, right? We also have the temperature that must be kept constant all the time, and also the carbon dioxide level, it must be kept constant all the time. So these 1, 2, 3, 4 are the factors that must be kept constant. So the examiner might not ask you about all of those, but they can choose maybe this factor, glucose, they can choose thyroxine level or salt level, either of those. So what you need to know, you must just understand what is negative feedback, so that should any of those be asked tomorrow, you are able to attempt that question. Okay, now we're going to look at the regulation of the thyroid levels in the human body. So what will happen if we've got low levels of the thyroid, the thyroxine? What will happen, right? So when thyroxine levels decrease, or when the thyroxine levels are low, what is going to happen? Pituitary gland is going to be stimulated, right? So when we've got low levels of the thyroxine that is secreted by the thyroid gland, what is going to happen? If it is low or if it decreases, the pituitary gland is going to be stimulated to produce more thyroid stimulating hormone. So it is going to produce more TSH, and then if we've got high TSH, that stimulates what? The thyroid gland. So the thyroid gland is going to be stimulated because of what? High levels of the TSH. Then what is going to happen? The thyroid gland will then secrete more thyroxine. Remember when we started, the thyroxine levels are low. So if they decrease or if they are low, what should happen? Pituitary gland is going to be stimulated to secrete more of TSH, right? Then the more we have that TSH, the thyroid gland is going to be stimulated to secrete more thyroxine. Then that brings the level of thyroxine to normal. Remember, it was high, it was low. Then for it to come to normal, what must happen? We need to have the thyroid gland secreting more thyroxine, so that the thyroxine level will increase. Then the thyroxine level is going to return to normal. Hence, we need the negative feedback that will work in opposite way, so that it brings the levels of those factors to normal, right? In summary, if we look at this Venn diagram, when thyroid level decreases, what will happen? The pituitary gland release more TSH, I'm summarizing that, and then the thyroid gland will then release more thyroxine. Then the thyroid level is going to increase and come back to normal. So it's important to understand how negative feedback works. Okay, right, now we are looking at the homeostatic control of carbon dioxide level. Yes, our body cells do not need too much of carbon dioxide. Okay, hence we are able to release it and take in oxygen. So we have that harmony of systems that are working together. So homeostatic control of carbon dioxide levels, because we don't need more of it, what is going to happen, right? So if we've got increased blood carbon dioxide, if we've got more carbon dioxide in the blood, then we'll be having decreased pH and also decrease in the oxygen level. We spoke about the receptors, the control center, and the effector. Let's look how they help in maintaining the homeostatic level here. So with the receptors, Remember, we've got low pH, we've got low oxygen, but increased blood carbon dioxide. What should happen there? We have chemoreceptors. Now, those chemoreceptors are going to be detected, right? Receptors, low pH is detected by chemoreceptors. Remember, we've got high carbon dioxide, right? So chemoreceptors are going to detect the low pH. Remember, the receptors are detecting what? The receptors are going to detect the imbalance of the factors, right? So now in the control center, the impulses then are going to be transferred, or impulses are going to be sent to the medulla oblongata. Remember, the receptors detected the imbalance of the carbon dioxide, which is high, right? So medulla oblongata is going to send those impulses where to the respiratory muscles and also to the heart muscles. So immediately if the impulses are sent to the medulla oblongata, what are we expecting? Because they are being sent to the respiratory muscles, we're going to have the diaphragm and the intercostal muscle increasing in the rate and depth of breathing. Hence, we are going to breathe faster there. And what is going to happen to the heart muscles, because it is impulses are also going to be sent to the heart muscles, then you are the rate of the heart rate is going to be increasing there. Okay, so when it is increased, let us look at the response. What will then happen? So the heart rate is going to increase and more carbon dioxide is going to be removed from the body. Remember, initially we had more of, or we had increased carbon dioxide in the blood, right? Now, in the response, in the negative feedback mechanism, or to bring the level of carbon dioxide to normal, what happens, right? Remember, impulses were sent to the respiratory muscles, where you are going to have an increase in the rate and depth of breathing. Then in the heart muscles, we're going to have increase in the heart rate. So when you're breathing faster in that way, what is going to happen? Heart rate increases, more carbon dioxide is going to be removed from the body. Then if more carbon dioxide is then removed from the body, because you had more of it in the body cells, now it is removed from the body, what is going to happen? Right, for negative feedback mechanism to happen, then blood carbon dioxide levels will fall. Remember, they were high here. The blood carbon dioxide was high or was increased. Then for negative feedback to happen, blood carbon dioxide level is going to fall, is going to decrease. Then the pH of blood is going to return to normal. So it's important to understand how negative feedback happens. Okay, so in summary, for the control of carbon dioxide in the blood, what we are saying is the carbon dioxide level in the blood is going to increase. The receptor cells in the carotid artery are going to be stimulated, and then impulses are going to be sent to the medulla oblongata in the brain, and then breathing muscles are going to contract more actively, and then increasing the rate and depth of breathing. And also the heart is going to beat faster, and as it is beating faster, more carbon dioxide then is going to be taken out to the lungs and exhaled faster from the lungs. Therefore, in that way, the carbon dioxide level in the blood is going to return to normal. That is negative feedback, right? We are going to look at the regulation of water, which is called osmoregulation, right? Now, with osmoregulation, yes, the body cells do not need more of that water. We don't need less of water. We can be dehydrated, by the way. So what is going to happen? let's say we have got normal water levels in the blood. So what is going to happen if you have got water level decreasing? All right, then the pituitary gland is going to secrete more ADH, right? Then more ADH, when it is secreted, we're going to have permeability of collecting ducts and distal convoluted tubules of the kidney increasing. Therefore, more water is going to be reabsorbed into the blood. Remember, it was less here. It decreased. Right. The opposite way of it, if we've got water level increasing, what is going to happen? If you've got more water level in your blood cells, the pituitary is going to stop secreting ADH. Remember, when we've got less of it, the pituitary secrete more ADH. But then when we've got more of water, pituitary stops to secrete ADH, or less ADH is going to be secreted. Now let us check what will happen to the kidneys there. We are going to have permeability of collecting ducts and distal convoluted tubule decreasing. Then when it decreases, we're going to have less water being reabsorbed into blood, or more water is going to be lost, and that will lead to what? Normal level in the blood. Normal water level in the blood. Remember, we're looking at the regulation of water, which is osmoregulation. So all those factors that we started with, or that I've shown you, must be kept constant all the time. Otherwise, what is going to happen if we've got more of it or less of it, Right. Now we're going to look at the negative feedback of what? The glucose. Remember, we get energy from the glucose. We get energy from the carbohydrates that we eat, the sweets. It can be those sweets that you took during your break time. Okay, so from the carbohydrates we get energy. Then what is going to happen if you've got, okay, the normal glucose level in the blood, it can be between the ranges of, it can be 3.9 and 7. Yes, we can say that. So the normal glucose level in the blood is acceptable at those ranges. Then what will happen if the glucose level in the blood increase? All right, so the pancreas is going to secrete insulin. Remember, glucose level is more, it has increased. Then what will happen? The pancreas as the organ is going to secrete what? Insulin, right? Then glucose is going to be converted to what? Glycogen. So we've got, do not confuse the glucose, the glycogen. We'll come to other terms that you know, right? Do not confuse all of those. So the pancreas is going to secrete insulin when we've got too much glucose in the blood. Then the glucose is going to be converted to a glycogen, and then glucose level is going to decrease and it comes back to normal. Then the opposite way, what will happen if the glucose level decreases? What will happen if the glucose level is low? Then the pancreas is going to secrete glucagon. Do not confuse glucagon with glucose and glycogen. Talk about those terms correctly. So, right, the pancreas secretes glucagon, and then glycogen is converted into glucose. Therefore, the glucose level is going to increase. Remember, it was low, right? It is going to increase. Then the sugar level, the glucose level is going to be, or it is going to come to normal again. It was low, now it is normal, right? How does that happen? Remember, don't forget, pancreas secrete what? Glucagon if glucose level is low, and then glycogen is going to convert the glucose, and the glucose level is going to increase, and it is going to go back to normal. That is negative feedback, right? We're also going to look at the salt, the sodium chloride. We need salt in the body. Yes, the body cells need salt, right? normal salt level in the blood, when it's normal, and what will happen if maybe you eat too much food that contains too much salt, or you're taking more of salt with your food that you're taking in, right? So when the normal salt level, when we've got normal salt level in the blood, what will happen should the salt level in the blood decrease, right? Remember, we are talking about the imbalances here. We need to maintain the constant level of all of this. The salt, for an example, here. So when the salt level in the blood decreases, what is going to happen? The adrenal gland of the kidney secretes more aldosterone. Remember, the salt level decreases, it's low. So adrenal gland of the kidney secrete more aldosterone. Then we're going to have reabsorption of sodium ions that are going to increase also, and the salt level in the blood is going to increase and go back to normal. Then the opposite way, what will happen if the salt level in the blood increases? What is going to happen? The adrenal gland stops secreting aldosterone. It stops to increase aldosterone. Remember, when the salt level is decreasing or is low, the adrenal gland is going to secrete aldosterone. But when the salt level in the blood increases, what we're saying is the adrenal gland is going to stop to secrete aldosterone, or it is going to secrete less of aldosterone. Then what is going to happen? Reabsorption of sodium ions into the blood is going to decrease. Then the salt level in the blood is going to decrease back to normal. So you must be able to, talk about the antagonistic way or the opposite way of maintaining the balance, the constant, or the balance, to balance the levels of the factors. It can be the salt, it can be the glucose, it can be carbon dioxide. That is negative feedback. Okay. Now, thermoregulation. Now, when we talk about thermoregulation, yes, we'll be looking at the temperatures. Okay. Now, with thermoregulation, we react differently in hot temperatures and also in cold temperatures. Right. Now, the temperatures, thermoregulation, we'll be looking at what the skin here. Right. Now, when we're looking at the skin, this is the structure of the skin. What we are saying, the blood vessels, when it's hot, in hot temperatures, blood vessels dilates. Okay, then more blood flows to the skin. More blood flows to the skin because blood vessels have dilated. That is in hot temperatures. And when it's hot, we know the sweat gland becomes more active and more sweat is released. So we release more sweat when it is hot, and then heat is going to be lost through what? Through evaporation. Okay, so what will happen in cold temperatures? When it is cold, we know blood vessels are going to constrict. So if you can even compare these two blood vessels there, right, they dilate when it's hot, and then when it's cold, the blood vessels constricts and less blood flows to the skin, right? So in cold temperatures, we've got less sweating, whereas in hot temperatures, we've got more sweaty, or more sweat is going to be released, and less sweat is going to be released when it is cold, right? Note that blood vessels do not move to the surface of the skin. Blood vessels do not move to the surface of the skin. But on a hot day, what happens? More blood flows in the blood vessels of the skin. It does not move to the surface of the skin, but on a hot day, more blood flows in the blood vessels of the skin. All right. So as you will be writing tomorrow, yes, Examiners might not ask about all those factors. Like I've mentioned, they can choose any one of those, right? Now, in this one, this is DBE May, June 2024. We're just going to quickly look at this question. And remember, given a question, you need to study what is given. We are being given a table here, right? Let's quickly look at the question. It says blood glucose level in a healthy person when not eating is between 3.9 and 7.1 liters of blood. The table below shows the blood glucose level in a healthy person who ate only one meal. Now, look at the question. Right, because we're talking about glucose level, you're thinking of what? Think of what? Homeostasis. Think of what? Negative feedback. Let's look at the question. So sometimes you don't miss the marks because you don't know the answer, but because maybe you did not answer or understand the question, right? So name the two hormones involved in normal homeostatic control of blood glucose level. We have already indicated that we've got insulin that is going to be secreted and also the glucagon, right? Now, name the organ in the human body that secretes hormone, that secretes insulin and also glucagon. Yes, we talk about the organ which is the pancreas. Remember, the pancreas has got isles of Langerhans that are going to be directly involved in the secretion of these hormones. Okay? Right? So it is the pancreas organ that is responsible to secrete insulin and glucagon. That is the pancreas. Okay? Now, understand the diagram given. Between which hours of the day did the person eat? So you need to look at the trend of the data that is given. So between which hours of the day did the person eat? So we are looking at 7, it was 4.2, and you can just look at any interval there. But between which hours of the day did the person eat here? Now, according to the graph that is given, it is from 8 to 9 because it was 4.2 and it increased. The blood glucose level increased to 8.4. So it is between 8 and 9. Give the evidence from the table. Give one reason for your answer that you provided in the previous question. So, using evidence from the table. So your answer should be based on the data that is given, not on your general knowledge. So if we say between these times, the person, okay, between which hours of the day did the person eat? We say between 8 and 9. Using the evidence from the table, give one reason for your answer, right? So the answer could be that blood glucose level has increased. From 8 to 9, it has increased. Remember, it was 4.2. At 9, it was 8.4. It has increased. So blood glucose level increased there. Okay? Right? And then, okay, let's continue with the question. Explain the changes in blood glucose level between 14 hours and 15 hours. Between 14 hours here. Explain the change in blood glucose level, right? Now, blood glucose level decreased below 3.9 at 2 at 14 hours, and then stimulating the isles of Langerhans or the pancreas to secrete what? Glucagon, which stimulates the conversion of glycogen. This is basically negative feedback, right? Which stimulates the conversion of glycogen to glucose, therefore increasing the blood glucose level at 3, right? It was 4.8, then 3.1, then 4.1. So it actually increased from 14 hours to 15 hours, from 3.1 to 4.1. Okay? Right. Describe how blood glucose level would have been different after 10. Right? This is 10. After 10, what happened? Describe how glucose level would have been different after 10 if the person suffered from diabetes mellitus. Okay? So what we are saying here, the levels would have remained high for a longer period if the person had diabetes mellitus. Okay. Now, because of time, I'm going to skip some of the slides so that we go to the next topic. Right? Plant response to the environment. So basically, with any topic that you're going to get, like my previous colleague mentioned, that you can be given scenarios, you can be given case studies, investigative question. So what is important, you should know the content of each topic. You should know the terms for each topic. So plant response to the environment. This takes 13 marks of paper 1. And then you should know the plant hormones. You should know the names. You should know that we've got auxin, we've got gibberellins, we've got abscisic acid. So what is important, you must know also their functions. What is their functions? Plant hormones. We have plant hormones here, the auxin, gibberellin, and abscisic acid. Right. Now I'm quickly going to look at the two here. Phototropism, the role of auxin in phototropism and geotropism. Now, what you should know, photo means light, and then tropism, growth. Okay, so phototropism is growth towards light or in response to light. Growth in response to light. So what will happen? Let's look at the two diagrams here. When auxin is produced, is secreted here, and light faces this direction of the where auxin is secreted. So because of light, auxin is going to move downwards. Auxin is going to diffuse down the shoot. Right now, remember, auxin is responsible for growth. So what is going to happen? The shoot here, the stem, is going to grow longer. Okay, now what will happen if light faces one direction of the plant? So this is the stem, and if light is from one direction, because auxin is sensitive to light, what is going to happen? Auxin is going to move to one side, right? And if it moves to one side, what are we expecting? The side that has got auxin is going to grow longer. So the shoot is going to elongate. So if one side of the shoot is in the light, auxin diffuses away from the light, then what is going to happen? So if this is unilateral light facing the shoot, we are expecting auxin to be on the other side, that is away from the light. Now, get into this one. If we've got light coming from this direction, and what we are saying is, because auxin is sensitive to light, it moves to the darker side of the plant. So the side of the shoot in the shade has more auxin. We're going to have more of it. Therefore, this side is going to grow longer. It's going to grow longer, right? So this will help. So growth take place faster here, causing the shoot to bend towards light. So we are saying the stem together with the leaves are positively phototropic, meaning they grow towards, or they grow in response to light, right? Let us look at the role of auxin in geotropism, growth towards gravity. So if you put your stem horizontal, what is going to happen, right? So if a plant is laid on its side, auxin gathers in the lower side. So when it gathers on the lower side, we are expecting what? Half of the stem and the root is going to grow towards gravity. Now, because it is accumulating on the lower side, what is going to happen? So the roots here are going to grow. Roots are growing towards gravity in this way. And then what will happen to the growth of the shoot? The stem is going to grow, because this side is going to elongate, basically because it has got more of auxin. So the shoot here, the stem is going to grow upward. What will happen? Because here, auxin slows growth in the root. So the root curves downwards. It's going to curve downwards, or it's going to grow towards gravity. All right? Right? I'm not going to go through the questions. I'm just going to explain what is in the diagram here. Right? Remember, when you are given the experiment, you can get if you get the aim of the experiment. We gave you examples when my colleague was mentioning that with investigative question, you must get the aim of the investigation or the experiment. And then in the aim of the experiment, you are able to get what? The variables, the independent, the dependent variable, the constant variable. You are also going to get factors that make reliability of the experiment or investigation and validity and so on, right? So because we are looking at the plant hormones check, so you are given the seeds here, right? Now, this is a developing seed. It grows a plumule, it grows a radicle. So we all know that from a radicals, the stem is going to grow. From a plumule, a stem is going to grow. From the radicle, the roots are going to grow, right? Now, experiment was set up to investigate the plant growth in response to stimulus. So a seedling has a radicle or a young root and a plumule, a young stem. This seedling was placed horizontal in a dark place and growth response was observed after 7 days. This is after 7 days, right? As shown in the diagram below, right? Name the growth response observed after 7 days. After 7 days, what do you notice there, or what do you observe there? We observe the growth response is geotropism or gravitropism, because the radical here is growing or is developing towards gravity. So the growth response observed here is geotropism, right? Plant hormone responsible for the growth response named here. We have got the auxin is responsible for growth. Do not forget that. And then explain the growth response observed in the root of the seedling, right? So if you are explaining, remember you need to indicate to show the cause and the effect. If you are explaining, we need to see the statement. We need to see the reasons also. So explain the growth response observed in the root of the seedling. We've got due to gravity, there is higher concentration of auxin on the lower side, okay, on the lower side of the root, which inhibits growth. Therefore, growth will occur mainly on the upper side, so causing the root to bend or to grow downwards. All right, I've got few minutes left, so you'll get this presentation from your teachers before you sleep, so that when you revise for tomorrow, at least you have the all the questions that I have prepared for you. Okay, so lastly, we're looking at the reproduction in vertebrates. Now, with reproduction in vertebrates, what you need to focus on, you need to focus on the following. You need to know the difference between external and internal fertilization. You need to know the reproductive strategies, the difference between ovipary, vivipary, and also ovovivipary. You need to know the amniotic egg. You need to know what is the difference between precocial and altricial development, and also parental care. What we mean when we talk about the parental care. So reproduction in vertebrates takes 8 marks of paper 1. So like we have indicated that, yes, with the 8 marks, it might be difficult question. It might be a simple question. So it is important to take all your work very serious. So in all the topic, know the terms under that topic. Okay, right. You should be able to compare the ovipary, ovovivipary, and also vivipary. Remember, you need to know how fertilization takes place between the three. You need to know uh, what type of fertilization is taking place, whether ovipary is external or internal, ovovivi is internal, and also the viviparous, it has got internal fertilization. Also, you need to know how the embryo develops. You need to know the development of the embryo. That in the ovipary, it is external to the body of the female. Ovovivipary is inside in the body of the female, and then with the vivipary is also inside in the female's body. All right, and also you need to know the type of nutrition for the three, right? For ovipary, for an example, the yolk is the only form of nutrition. The yolk is the only form of nutrition, right? Developing the yolk is the only form of nutrition for the developing embryo. And then in the ovipary, we're looking at the nutrition. Remember, the yolk present in the egg, young are independent of the mother's body. What about the vivipary? Young receive nutrition from the mother's body through the placenta. We have looked at human reproduction there. You have looked at how the placenta is formed, stuff like that. So vivipary, we having what? Young receive nutrition from the mother. Okay, type of the egg between the three. Type of the egg of the ovipary is jelly-like, is calcareous, and then ovipary is calcareous or leathery also. And with the vivipary, they don't have the eggs. We've got none there. Okay? Right. I believe that you have dealt with many questions from past papers. So this is one of the questions that was asked in November 2023. That is last year DBE, right? So I'm looking at my time, but what we are saying is, okay, let's just attempt this one question. This is the last slide, I believe, right? In some frog species during mating, the male climbs into the back of the female and grabs her with his front legs. During this time, the female will release about 6,000 ova, while the male releases sperm onto them. This mating behavior is called amplexus. Remember, you can be given case studies, even if you have not studied that, but given a case study, read that, because some of the questions are going to be based on that case study, right? 211. The type of fertilization that occurs during reproduction in frogs. Remember, we said fertilization can be internal, external. It depends on the organisms there. So in the frogs, name the type of fertilization. It's external, right? Explain why the fertilized egg of the frogs do not survive on land, right? So you can just think about that question. Remember, you need to know the suitability of organisms in terms of where they stay, where they live, where we find them. So if we look at the frogs, explain why the fertilized egg of these frogs do not survive on land. What do you think? All right. The eggs will dry out because it's on land, because they have no shells again, and they are amniotic eggs, and they have no amnion to protect those. Right? Then explain how amplexus increase the chances of fertilization in frogs. Remember, with your explanation, show the cause, show the effect. So the male and female bodies are in close contact, right? As you can see, so that sperm can be released directly into the ova, or according to the given information here from this data, many or up to 6,000 is in the information is in the case study that is given. Up to 6,000 ova are released since fertilization is external. So you can get your free marks there. All right. So before you sleep, make sure that you look at practice. Check yourself. Check yourself, what you know, what you don't know in preparation for tomorrow. Just attempt different questions. Remember, right now, yours is to practice and practice and practice. We are left with few hours, like I've indicated. Okay? Not even 24 hours. We left with few hours for you to prepare. So in order for you to be prepared, right, you look at the notes that you were using, the one pager that we were giving you, practice. Maybe you can just take one set of question paper to drill yourself tonight so that by tomorrow you are ready. I want to wish you good luck as you'll be writing your paper one tomorrow. And I believe that you are going to make it. I want to also wish you good luck as you are going to be writing your paper 2 on Monday. Remember, tomorrow after writing, you are going to write up to 11:30, and then 2 o'clock we're going to have the very same session to revise, to revise on your paper 2 work. Good luck and goodbye. Thank you.